Thank you for honoring my father, Dr. Saul Hertz, who conceived and brought from bench to bedside the medical uses of radioactive iodine, RAI. He'd been using stable iodine in X-rays as a non-surgical method to treat goiters, as surgery was dangerous and expensive. A eureka moment occurred on November 12, 1936, when Dr. Hertz spontaneously asked MIT President Carl Compton, could iodine be made radioactive artificially? Harvard funded the initial research. MIT Rad Lab director hired physicist Arthur Roberts, PhD, to work with Hertz. Roberts produced non-cyclotron I-128. Hertz and Roberts successfully demonstrated the use of RAI as a tracer to study thyroid physiology. The director of the Rad Lab did no work related to the research. It is documented that the director forced his name on the author's list once the paper had been approved for publication. Correspondence verifies the director had difficulty working with Hertz and Roberts as they were outsiders. Importantly, in 1937, at the time of the preclinical studies, Dr. Hertz conceived of RAI to treat thyroid carcinoma. How do I get the next slide? I don't know how to do it. Somebody's going to have to show me. Sorry, this is going to take
Just
a
the
while.
green button. Just
Okay,
the green
got it. Thank you. On March 31st, 1941, Dr. Hertz rushed over the Charles River Bridge with a flask of MIT-produced RAI. He administered the very first therapeutic use of an artificially produced radioactive substance. Uptake testing and dosimetry were used to determine a safe, effective dose. In 1942, he conducted and reported his trials of cancer patients with thyroid cancer patients to the Markle Foundation that funded the MIT cyclotron. By the way, it was $30,000 to buy a cyclotron in the day. Such a deal. In 1943, before leaving for World War II, Dr. Hertz advised New York Dr. Seidlin in the use of RAI to treat thyroid cancer. Patient BB arrived at Montefiore Hospital in New York and had had his thyroid surgically removed 15 years earlier. BB was treated with RAI as directed by Dr. Hertz. No new lesions appeared, and some completely disappeared. However, in 1952, BB succumbed to anaplastic carcinoma. I think I may have missed a slide here. Okay. There he is. That's BB. After the war, Hertz's military leave of absence was not honored at MGH. His replacement at MGH went to publish Hertz's work as his own, omitting both Hertz and Roberts. In 1946, with funding from the Navy, Dr. Hertz refined the use of RAI to diagnose and treat thyroid carcinoma at Boston's Beth Israel Hospital. Hertz stated, "My new research project is in cancer of the thyroid, which I believe holds the key to the larger problem of cancer in general." In September 1946, he founded the Radioactive Isotope Institute with facilities in Boston and New York. In 1946, in June, he's quoted as saying, "Demand is expected for radioactive iodine, and as research develops in the fields of cancer and leukemia, for other radioactive medicines." He successfully treated thyroid cancer and researched the use of radionuclides and hormones for other forms of cancer. A May 1949 Harvard Crimson headline read, "Hertz to use nuclear fission and cure for cancer." The cost of radioiodine was dramatically reduced with the production of RAI from the atomic pyres. In 1949, Hertz established the first nuclear medicine department at the Massachusetts Women's Hospital, reported as opening a new division where radioactive isotopes will be used to study and treat disease. Hertz stressed the tracer-targeted approach. With tenacity, courage, and passion, Saul Hertz overcame the racism of his time, a world war interrupting his work, the establishment pushing back, and the fear factor to bring his work to fruition. A cancer survivor emailed, "The cure delivered on the wings of prayer was Dr. Saul Hertz's discovery, the miracle of RAI. Few can equal such a wonderful and precious gift." Yes, our next speaker carries the legacy forward with more than 1,600 published papers, 1,400 patents, and is co-founder of Moderna. So please welcome MIT Institute Professor Robert Langer.
Well, thank you all for coming, and it's really an honor to be able to follow up Barbara and to really, you look back and you see the enormous impact of what Saul Hertz has done. I asked Barbara what she wanted me to say. She sent me this, I just took it right from her email. So that's how I've built on the foundation, which is a remarkable foundation. Training great people and establishing companies. So I'll just be very brief. My background, I got my degree in chemical engineering in 1974. And when all my friends went to work in the oil industry, I wasn't that excited about that. And I took a job actually at Boston Children's Hospital, by far the lowest paying job, but with a man named Judah Folkman, and who was a surgeon. And I was the only engineer in the entire place. But Dr. Folkman asked me to try to help him solve a problem, which was he had a theory that if you could stop blood vessels, that might be a whole new way to treat cancer someday. And he was widely criticized for that theory. But, and so we had to develop ways to study blood vessels. There wasn't any way to do that in 1974. And so the way we did it is there's certain sites that don't have any blood vessels, and we put tumors there, and then we had to create tiny little nanoparticles that could deliver large molecules, which had never been done before. And at that time, everyone said that that was scientifically impossible. Dr. Folkman and I hadn't really read the literature saying that, so we tried anyhow. And I actually worked on this for a long time. I found over 200 ways to get it to not work. But eventually, we did get it to work. We published 2 papers, one in the journal Nature in 1976, showing for the first time you could create tiny particles, like a nanoparticle, that could deliver large molecules, including even nucleic acids, which RNA and DNA are. And one in Science, also that same year, using this approach to actually isolate the very first angiogenesis inhibitors. So what happened over many years are a few things really done by many people, but where those 2 papers and what we did and what I tried to do subsequently would make a difference. The blood vessel inhibitors. So I told you our paper was 1976. It took 28 years from that paper before the first angiogenesis inhibitor would get approved. But starting in 2004, something remarkable happened. Avastin gets approved. Avastin is the number 2 or 3 best-selling biotech drug in history. And one after the next blood vessel inhibitors are approved. Even last year, it was the 4th biggest selling drug in the world. And not only are these drugs widely used for cancer, Nature, the journal estimates 500 million people will be treated with these. These are really the only treatments, pharmacologic treatments, for diseases of blindness in the back of the eye, like macular degeneration or diabetic retinopathy. These are drugs like Illya and Lucentis. The drug delivery work, well, there I thought, gee, we'd done this work. I was so naive, I thought people would build on it. But really, I got large companies involved for a little bit, and they kind of gave up. So I got the idea of starting companies myself. I kind of learned if you're not your own champion, nobody else will be. So we started companies over the years. That also led to many different products. Probably the best known of them is this one, which some of you may have had. That's the COVID vaccine from Moderna, which I was able to start with Nubar Fayan, Ken Chen, and Derek Rossi. But there are many, many others there that we've helped on, and like everything from drug-eluting stents to new treatments for cancer, schizophrenia, and many others. And then the one other area that we started, and again, all my work is at the interface of kind of materials and medicine, is that you could someday, maybe you could make organs or tissues from scratch. And that would lead, on this last slide, to everything from new skin for burn victims, actually blood vessels, which Laura Nicholson, one of my students, started a company on, Humacyte, and that actually got approved last December, and they're actually used quite widely now in the Ukraine for treating wounded soldiers. And but not only can you do these for all these tissues, we're also working on organs and tissues on a chip. And of course, the most recent one and the beauty, I should say, of organs and tissues on a chip is if you try to do human testing, it doesn't go real fast. It's hard to control everything. If you try to do animals, well, animals aren't people. And so, but if you could do it, really make authentic systems on a chip, that might give you an opportunity to find new drug targets and so forth. And working with Li Wei Sai, this wonderful postdoc, Alice Stanton, and who's now a professor at Mass General and Harvard, who's actually here, this just came out a couple of days ago, she's actually made a brain on a chip where you can actually, and these are personalized brains. She can take any of your cells, convert them into what are called iPS cells, and convert them into the 6 different brain cell types. And she found a matrix, so they'll reorganize, and they make mini brains. At any rate, we're working on a lot of these, and that's what we try to do. We're also doing a lot of work with the Gates Foundation to try to bring all these new technologies to the developing world, including better nutrition and so forth. So I'll maybe stop here and see if I can answer a question or two before we go to the next talk. But I'm really honored to be able to present something, in the honor of somebody who's done the remarkable things that Saul Hertz has done. Anyhow, any questions? Okay, well then maybe we'll continue. So then the next presenter is Illya Gipp, who's at GE Healthcare, medical officer for oncology, and he's going to talk beyond the half-life, linking a discovery of the past to the future of cancer care. Sure.
If I can get my slides, and thank you for the sound. Yeah. Now, I do represent GE Healthcare. I'm the chief medical officer at the company, but I'm not going to be talking anything commercial. So this is not a commercial event, and I really try to get the title really beyond the half-life and linking the discovery of the past to the future of cancer care. Now, where I would like to start is the breakthrough innovation that took place 84 years ago. There is a picture right here. I think everyone in this room is familiar with what this picture is about. But I think that there is more than just the radioactive iodine. And if I were to summarize on what I really consider important regarding the real breakthrough, in my view, is number one, this, to my knowledge, was the first targeted approach in medicine. Now, if I go right now and Google targeted cancer care, the first thing that pops up is 1970s and tamoxifen and breast cancer. But really, the targeted approach, because of the ability of iodine to reach thyroid, was really pioneered by Dr. Saul Hertz. Number two is this was really one of the first approaches toward what we call today precision medicine. Realization, assessment, and response prediction, because that device that is also shown here and that is being touched by Dr. Saul Hertz is something that enabled clinicians not only to inject and administer, but also to see how the distribution took place and if this really has the therapeutic effect. Number three is that this was indeed the first radionuclide therapeutic use. The first radiotheranostics concept, because, well, maybe at that time it was not really necessary and there was no equipment that allowed people to really visualize. There was no SPECT CTs. But through iodine 123 and iodine 131, this can really be considered the pair that can be used for diagnostic and for therapeutic applications. And even used today, maybe in some challenging cases for thyroid cancer, but definitely going into MIBG. And the last but not least is it laid the foundation for personalization and adaptive therapy strategies, again, with the fact that Geiger counters was used to assess and measure the effects and the distribution of the radioisotope after it was administered. So going on further, it really took off, but it took off in an interesting manner, because the first publications that came out actually from Montefiore in New York, Dr. Sadlin, he consulted with Dr. Saul Hertz, and he understood that this is where it can really give a further advancement into cancer care. And indeed so just a few years later, you can see that things started to be quantified, things started to be objective, and started to be actionable so it could really be populated throughout the medical community. Now, this is something I wanted to brag about, and this is actually a part of what I believe was a beautiful coincidence. Because at that time, the more known isotope was iodine 128. Right, discovered by Enrico Fermi quite a few years earlier. Well, the point is that at that time, it could have been used because it also has a therapeutic potential through the beta decay. But the problem was that it is having a much shorter half-life. What I did today in the morning is I did the run, and I did the run from the original building where the cyclotron was when the iodine 131 was generated to the Mass General building, or right next to that building, where it was administered. So I called this the first activity run. But the point is, I am not nearly as good of a runner. If this was iodine 128, none of the activity would have survived by the time that I would have gotten to Mass General. So the fact that it was practical, the fact that it was measured, it really laid the foundation for something that we're using today. And it's being used quite intensively. Now, I looked for some of the papers that are available today, and you can see that approximately 10,000 people of all ages are treated with radioiodine each year in the UK alone. That's their NHS data. So we're talking about hundreds of thousands and millions of patients being treated today. So something that started 84 years ago is with us today. There is also a lot of anticipation regarding radioactive iodine ablation therapy market forecast to continuously grow by approximately 5%, which I think we all would agree is not bad. And the most important is just recently, I think 2 or 3 years ago, the review paper came out on the strategies for radioiodine treatment, and it talked about the fact that radioiodine treatment represents the most widespread and effective therapy for differentiated thyroid cancer. Quite impressive for the targeted therapy that pioneered in 1941. So I think the legacy of this meeting is really talking about something that is so important that it has the future. Now, as of today,